Jikomis, and today I'm speaking with Dr. Dave Manoli. Professor Manoli is a professor at the University of California at San Francisco, where he is both a clinician and a researcher, a neuroscientist. On the neuroscience side, his lab studies the neurobiological basis of behavior. In particular, they focus on social behavior and attachment and how these things are shaped by experience, how they are related to neuropsychiatric disease, and they study a really interesting model organism called the prairie vole, which is one of the few socially monogamous rodent species out there. They form long, sometimes lifetime pair bonds between the male and the female. Both parents invest a lot in the offspring, and their sort of mating and reproductive behavior in many ways actually looks a lot like human beings, another monogamous species. On the clinical side, he actually works at the UCSF Early Psychosis Clinic, where he treats patients with childhood psychosis. And so he thinks a lot about how the brain, creates our ability to form social attachments, how behavior is governed in general, but in particular, things like parental behavior and mating or sexual behavior and social behavior between individuals. And, how is the brain orchestrating all of those things? What are the neurobiological and the neurohormonal inputs that sort of sculpt how those things develop in the brain and how behavior is governed by neural circuits in the brain? So he thinks a lot about how the basic research that his lab conducts connects with his work on the clinical side. So that has to do with human and childhood psychiatric illness. We talked about a lot of interesting stuff. We talked about, why some species are monogamous and why some species are promiscuous. We talked about parental behavior and care behavior and how parents and offspring become attached to each other. We talked about how males and females become attached to each other in the courtship process. We talked about, defensive and aggressive behaviors and affiliative behaviors. We talked about things like oxytocin and other hormones like testosterone and estrogen and how those things influence how the brain develops, how some of these social behaviors develop. We talked about everything from menopause in females to in-group versus out-group distinctions. We got into a little bit on how the olfactory system works, how animals can smell things that are floating through the air or smell things like pheromones that are present on other members of their species. We talked about how different parts of the brain express or are sensitive to things like estrogens and testosterone and oxytocin. We got into some of the details on how those things work at the molecular level and the neural level. And towards the end, we tied some of that into the human side. So neuropsychiatric diseases like childhood psychosis, how things that happen early in development can shape how you respond to things or how you behave and perceive the world later in life. And so if you're interested in behavior, broadly speaking, but especially if you're interested in social behavior, that's mating and sexual behavior, parental behavior, all of that stuff. How do we think about that in terms of neurons? How do we think about that in terms of ecology and evolution and why things have evolved the way they have in different animals? This is a really, really interesting episode that covered a lot of ground, all the way from the molecular genetics up to the level of psychiatry and psychology. As usual, if you enjoy the content I'm producing, please like, share, and subscribe. Don't forget to check out mindandmatter.substack.com, as well as the links in the episode description if you want to learn how to support the podcast further. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. Hey everyone, I want to take a minute to tell you about a product I use called Everyday Dose. They have created excellent coffee and matcha products with functional mushrooms and other supplements and less caffeine than traditional coffee or matcha products. I actually reached out to them because I've been using their product for about a year or so, and listeners often ask me about my daily and weekly diet habits. They make a really good mushroom-based coffee alternative. It contains micronutrients with antioxidant and anti-inflammatory properties, as well as collagen protein to help support healthier skin, nails, hair, and joints, and the amino acid L-theanine from tea leaves. Each cup has just about 39 milligrams of caffeine. That helps eliminate the caffeine crash that can come if you drink regular coffee, which has much higher caffeine levels. And they use a unique cold extraction process that results in lower acidity than normal coffee. And the caffeine microdose makes it suitable even for someone who doesn't normally drink coffee. This mushroom-based product is made using a double extraction from 100% mushroom fruiting bodies like lion's mane and chaga to maximize the extraction of micronutrients like beta-glucans, triterpenes, and sterols. Other brands don't typically do this, making Everyday Dose one of the highest quality products of its kind. It's gluten, dairy, and nut-free. There's no added sugar. It's paleo and keto friendly and made with kosher ingredients. There are no grains or fillers, and it is lab tested to ensure quality. I really like the taste of Everyday Dose compared to black coffee and other mushroom coffees, and they have a mushroom matcha product loaded with functional mushrooms and collagen proteins. So if you like green tea matcha, you'll probably like that product too. If you're interested in a healthy coffee alternative, I highly recommend giving Everyday Dose a try. Check out the link in the episode description or visit everydaydose.com to learn more. If you go there, you can find special offers that they have for getting a free frother and free travel pack of on-the-go doses with your purchase. And with that, here's my conversation with Dr. Dave Manoli. Everyone a little bit about who you are and what your lab studies generally?
Sure. So my name is Dave Manoli. I'm an assistant professor in the Department of Psychiatry and Behavioral Sciences at UCSF. I'm mostly a basic neuroscientist, and my lab works on the neural mechanisms underlying social attachment, everything from sort of the basic molecular biology at the individual cellular level for groups of neurons involved in how we build social memories, up to how these circuits function to guide behavior. I'm also a child psychiatrist, and my sort of clinical area of specialty is in early psychosis, and understanding social deficits that can occur in the context of a number of neurodevelopmental syndromes, but also how they may influence or potentially even predict the onset of psychiatric disorders later in adolescence or in adulthood. So yeah, I think that probably covers.
All right, so you're a psychiatrist. You actually see human patients. You also run a basic neuroscience research lab. Is that a mouse lab? What kind of animals do you study?
So we use a mouse-like rodent called the prairie vole. That's just about the size of a hamster, so it's a little bit bigger than the sort of average laboratory mouse. And the special thing about prairie voles is that they're in the sort of 3 to 5% of mammals that are what we call socially monogamous. And so what that means is that these animals form lifelong bonds between mates, and that this is something that endures throughout adulthood. And another correlate to that is that in addition to actually wanting to spend time with each other, there's a really profound change in their behavior with regards to other potential mates, and that they completely reject novel potential mates. So you can imagine there's lots of interesting both neurobiology as well as behavioral biology about how these sorts of adaptations to social behavior have evolved, how they occur in the brain. And then one of the other sort of convergences between sort of my clinical interests and our basic science lab is how are these actually influenced by either the genetics associated with neuropsychiatric illnesses or early life experience? How does that either facilitate sort of stronger relationships later in life, or how does it actually disrupt the formation of these, when these animals grow up, or if we're talking about people in the context of early life experience?
Yeah, I definitely have a lot I want to dig into with respect to the prairie voles because they're really interesting, and they're also sort of a less commonly studied lab animal. So there's really a lot of interesting stuff to dig in there. Before we get to that, I wanted to just sort of set things up with some really basic questions. So, as a neuroscientist and a neuroethologist, someone who studies animal behavior, can you just define for everyone how someone like you would define animal behavior in general and social behavior in particular?
So, I mean, I think the general term animal behavior is really sort of any observational pattern of activity at the sort of fine scale level when we're talking about sort of simple sequences of movements that sort of comprise what we call a behavioral sequence, to large scale interactions. How does an animal respond to a specific change in its environment, to a specific stimulus? So that might contrast sort of spontaneous versus evoked behavior at a very, very macroscopic level. And then when we go into the realm of social behavior, I think at the most liberal definition, it's really how do animals interact with each other, whether we're talking about what we call conspecifics of members of the same species, or, for example, if you were to take an animal and its predator or an animal and its prey, interspecific interactions. And I think all of those really form sort of this context of social behavior. And maybe if it's okay for the purposes of the conversation, we can say social behavior is largely sort of restricted for the purpose of the conversation to within the species. But I think, especially when it comes to attachment, and this may be a direction we go in, there's really interesting intersections between how animals attach to each other. And, for example, with certain animals that we come to love in our homes, how they attach to other members or members of other species.
Yeah, no, I can relate to that. I'm actually at my grandma's house right now, and and so there's a new puppy here, and so we all know how that goes.
You went where I was going, right?
Yeah. Well, you actually wrote, I think you wrote like a news and views for a recent study, and you were talking about like the context dependence of behavior. Our behavioral tendencies change throughout our lifespan. There's developmental processes that go on when certain things happen to us. We go through puberty, or we have babies, or whatever. Our behavior patterns can change, and I think you actually used the example of female elephants to sort of talk about how behavior can really change in quite dramatic ways depending on the external and the internal context of the animal. Could you maybe talk about the elephant example a little bit, just to get people thinking about how behavior changes across the lifespan and in different contexts?
So, I mean, I think that, really that sort of reflects sort of how hormonal states, for example, or different external stimuli change the state of the animal. And so you can imagine that if we're talking about sort of a fear response, you're going to have a very, very different set of behaviors in the same context than if you're talking about, for example, the display of aggression in the context of territoriality. Or if we're talking about nurturing behavior, right? If we're talking about a mother nurturing its young versus a mother defending its young against a threat, right? And so in the context of each of those sort of defensive behaviors, you're actually sort of seeing similar patterns of behaviors displayed in different ways and in different contexts that are really dependent upon the context of the animal, that's both sort of influenced by external stimuli, but sort of encoded from the internal state of the animal.
And so like when we think about, everyone can think of examples in their lives of like when their behavior changes for like purely internal reasons, right? So you can imagine, we all have examples in our own lives where, we're in the same basic context at, time point A in our life. And then at time point B, we're in that context again. But our behavioral response is very, very different. Mm-hmm. One of the things that sort of precipitates some of these changes are all of the hormonal and neural changes that come with, the onset of puberty and becoming sexually mature. Another one is, having children, or when an animal has babies. So how do we start to think about what's driving those changes? Things like, sex hormones and other factors that are circulating throughout the body, and how it is that they actually start to change behavior.
So I think in the context of sex hormones, it's worth taking a little bit of a step back in terms of how we think about the actions of sex hormones on the brain. So the general sort of model, especially when we're talking about mammals, for example, is what we call the sort of organization and activation model. And the organization part of it is very, very early on. And, for primates, it could be actually even intrauterine, probably also for some very, very small aspects of rodent development. But then very early on, you have hormones that differ, right, between males and females in different ways. And as a consequence of that, those hormones have actions in the brain. And where those actions generally occur are where there are specific receptors for these hormones. For example, testosterone, estrogen, right? They have individual types of receptors that are in not everywhere in the brain, but in specific regions. And so they have developmental consequences. They can wire the brain differently, or at least change the physiology or even the number of neurons in different parts of the brain. In general, when we're thinking about these sort of innate behaviors, we think of the hypothalamus, or like sort of the most sort of basal reptilian brain, as it were, that are the most dimorphic or the most different between males and females. And they're organized during this early developmental period. But then there's generally a period where kind of the hormones aren't around, and this is sort of pre-puberty. And then the beginning of puberty, we start to see these hormones come up. And that's where this sort of activation aspect of the model comes into play. That, not only are these circuits now different from a developmental standpoint, but then they're sort of primed or activated to elicit or to at least potentiate different types of behaviors. And an important feature is that there is probably a lot of wiring that is involved in the behaviors that, for example, differ between males and females that are common, and that the certain subsets of neurons that are really different influence the probability or the likelihood of displaying one or the other. But for the most part, both animals of a species, if we're talking in general about mammals, can do the same sorts of things. They just do it in different contexts. And so that's where the really big difference when it comes to adult social behaviors really manifests. What is the difference between when a female mouse or a mammal experiences estrus and then is induced to be reproductively receptive versus, for example, a male who then has testosterone is induced to mate, with the sort of typical displays of mating behavior that they engage in.
Mm-hmm. And can you talk a little bit more about what most people think of as the two main sex hormones, testosterone and estrogen, and in particular, the way that people talk about these outside of technical fields is testosterone is the male hormone, estrogen is the female hormone. To what extent is that an oversimplification? And where do these hormones come from? How many of them are produced solely by the gonads? And to what extent are some of these things actually made and deployed within the brain itself?
So I think that I'll go to your first point about, the difference between testosterone and estrogen. And so basically, testosterone is a precursor to estrogen. And so the simplification of testosterone male, estrogen female is an oversimplification in a number of different ways. And I'm going to focus just more on the sort of behavioral or the neurobiological component. And there's going to be an important distinction between what potentially is happening in primates versus what we've sort of elucidated happens, for example, in mice. But what we found, or the field found, is that estrogen is actually one of the principal masculinizers of the rodent brain early in development. And what happens is that testosterone is produced, and it is locally changed into estrogen in different parts of the brain. There's a specific enzyme that does this that is also very, very restricted. And by virtue of that, circulating testosterone enters the brain, becomes estrogen, and then estrogen works on different parts of the male brain to masculinize it. In females, there really isn't circulating estrogen. There's not circulating testosterone because they don't have male gonads. And so there's, in general, what we would say is sort of a default differentiation. That's a very, very gross oversimplification, but there is a pathway that happens in the absence of this sort of early estrogen signaling that is sort of the differentiation of the female brain. And so it was very paradoxical when sort of researchers were trying to manipulate these neurons or these hormones and actually identified that males that don't respond to estrogen genetically actually were demasculinized. And that sort of gave rise to this aromatization hypothesis of how estrogen is actually the masculinizing compound. Now, in primates, there's a little bit more of a difference that happens in terms of how things are occurring in utero. There's also an expansion of the number of neurons and the types of neurons that respond to androgen or testosterone via the androgen receptor. And so there's probably a little bit more, not surprisingly, more nuanced differentiation, both of males and females for primates.
I see. So the reason we can't really just think of testosterone equals male, estrogen equals female, is because even in males, when you have testosterone circulating throughout the blood, it goes into the brain. But then in very specific locations, it sounds like you can, in some parts, in some cases, turn that testosterone into estrogen, and it's actually having a masculinizing effect, making the circuits more male typical or whatever.
More male typical, right. And actually another important thing that is sort of happening at the same time is that actual process is what actually induces the expression for testosterone, of the receptor for testosterone in certain parts of the brain. So estrogen really starts first, and then there's this whole pathway that it then sort of potentiates this sort of cycle of differentiation by virtue of causing more places to respond to testosterone.
I see. So you can have the induction of testosterone receptors by estrogen itself, which then subsequently makes new neurons and new circuits responsive to testosterone later on.
Exactly. And you can imagine with this sort of very nuanced sort of genetic programming in primates, which have a lot more differentiation happening in utero, right? You have actually access to different hormonal surges. And so that balance gets spread out over a much larger period of time. And there's more complicated nuances because of sort of the genetic interactions between estrogen and testosterone signaling in different parts of the brain.
Mm-hmm. And so, I guess for mammals on average, I guess one question would be like how general are some of these things? How species specific are they? The difference between males and females, one way to characterize that it sounds like is there's a different pattern of waxing and waning of the sex hormones at different phases of development. Is that how we can start to think about it?
So I think that I would say that that's sort of maybe the conductor for sort of these processes that, to a certain extent, there's part of the score that's been written by the same players earlier on, and then the phases of the hormones sort of say, okay, what are we going to do in terms of differentiating the score versus what are we going to do in terms of how it's going to play out, right? So you have these early players that sort of, again, this organization, and then depending upon whether you've differentiated in a more male typical or a female typical way, how the hormones influence what's happening in sort of adulthood or adolescence, then that's dictated by these fluctuations. And as you sort of alluded to, even the hormones themselves have different patterns of fluctuations, right? The sort of the estrogen or the estrus hormones go through their own cycle, versus testosterone in males tends to have a much more frequent or higher frequency of cycling.
Interesting. And to me, one of the keys so far is that the pattern of hormone flux across development actually sets up how sensitive circuits will be to some of these hormones later on. Yeah. And so I would imagine there's some pretty big developmental effects there where, if something happens or doesn't happen at one phase of development, it's then necessarily going to cause some later phase to change how things manifest.
So the metaphor that's often used is that it sets up the sort of genomic landscape, or it regulates different parts of the genome to then be responsive to these later hormone surges, right? Because the receptors that we're talking about actually act on DNAs once they bind these hormones. So they're what we call transcription factors, and they dictate both changes in gene expression, but also changes in overall genomic structure. So, places that respond to a hormone early then are primed to either respond or not respond to hormones in a specific way later on in life. And so that also dictates not only how neurons function, but how genes actually are induced or repressed in response to the hormones in adults.
I see. So when we're thinking about the effects of sex hormones on behavior and how that happens at the molecular level, hormones actually get into the brain, physically go inside of neurons, and they change, genes coming on and off and all that stuff.
Yeah. And then the one actual tweak to all of this is that, for example, with estrogen, just to make things even more complicated, there's even a receptor that's not one of these transcription factors that's, for example, at the surface of a cell that actually changes the physiology of cells in response to estrogen. So there's many, many timescales in which these hormones are affecting a lot of different processes that sort of dictate how a neuron is going to function or respond to other stimuli.
Interesting. And how sensitive are patterns of hormone flux to like environmental inputs? What the animal's doing and what's happening to it at different phases of development.
So it really depends on sort of what are the mechanisms that are transferring those signals from the environment into the brain, right? So there are certain hormones, for example, like the ones that, eventually I think will lead to like oxytocin and vasopressin, that operate on potentially larger timescales, although they have effects in neurons very acutely, and that influences sort of the timescale of their responsiveness. But for example, when we're talking about the steroid hormones, the gonadal steroid hormones, they have actually overlap directly in terms of some of their receptors with stress hormones, like the glucocorticoids. And so those are operating now on a timescale where we're dramatically influencing patterns of gene expression that aren't happening at like sort of the millisecond timescale, but over, minutes, hours, days, even longer than that. And so you can imagine, depending upon the developmental context, for example, if we're talking these early developmental stages, if there's higher levels of stress hormones that are influencing how the steroid hormones can actually even get access to parts of the genome, that can have a very, very dramatic effect that has developmental consequences long term, right? Versus, for example, later in life, there are stress hormones, obviously, like glucocorticoids, but there are other pathways that influence the dynamics that these circuits can actually respond to, or how they're responding to other stimuli, whether they're sex hormones or other signals.
Mm-hmm. Yeah, and so it would seem obvious then that, depending on what happens to you during development, for example, if you have relatively high levels of stressors versus low levels of stressors, all of these things are going to interact, and they're probably going to have like cascading effects on how, subsequent sexual behavior, social behavior, anxiety levels, and, almost everything you can think of will sort of manifest later in life.
That's perfectly articulated. That's exactly, and that's one of the things that we're very interested in particular, how it influences sort of the social and attachment aspects, because, I think we know as people, but also there's lots and lots of animal literature that those early experiences have very reproducible and profound effects. So the question is, how does that actually happen at a sort of molecular and circuit level?
Mm-hmm. And you mentioned earlier that you studied prairie voles, and I don't think everyone knows this, but I think there's probably a good general sense out there that obviously not all animals are monogamous. There's a range of sort of promiscuity with which animals display sexual interactions with conspecifics, with members of their same species. Some are very promiscuous and they sort of mate, and there's no real attachment that forms between the male and the female beyond that. And then, humans and prairie voles, I think, would be, sort of one extreme where we're much more monogamous than the majority of other species. What was that number that you cited for how frequent monogamy is in the animal kingdom?
So that's actually, the earliest estimates were from a researcher named Devanand Manoli, who in the late 70s basically estimated about 3 to 5% of mammals. But even within mammals, right, there's a huge diversity. So as you mentioned, with primates, there's a slightly higher than average, than sort of across mammals, average of monogamous mating systems. But if we go, for example, to canids, right, there's actually like, I think probably now 3 or 4 year old sort of study / review, where in the wild, with the exception of domesticated dogs, you do not see canid species that don't display some form of social monogamy, sort of independent of their complex social structure. If we go to like songbirds, outside of mammals, 90% of songbirds are monogamous. And so there's huge variability in terms of both just sort of the patterns of mating systems, and then also the sort of the likelihood that a species is going to actually display those types of behaviors.
Mm-hmm. And when we say monogamy, I'm curious to have you define what exactly that means in two respects. So one, when we're talking about sexual behavior, does monogamy mean lifelong pair bonding? Does it mean for a season or like some quote unquote long period of time? And then does this only refer to sexual behavior? Can we talk about social monogamy in a more general way?
So I think maybe for the purpose of the conversation, I would sort of veer into the realm of social monogamy, right? Because in most systems, you see that if you really look at the patterns of behavior, you see sort of what we'd say extra pair matings, right? And they happen with different frequencies in different contexts. They're actually influenced by different environmental contexts. And so in general, we're talking about sort of the social monogamy that's displayed by a mating pair. And as you sort of alluded to, there are certain species where it's lifelong, and then there are other species where it's basically a reproductive cycle, and that varies. Again, the frequency and the sort of the styles sort of vary across different subsets of species. Presumably based off of sort of the evolutionary pressures that it's evolved in.
Yeah, I mean that's actually an interesting question. Is do we know anything about what some of those evolutionary drivers are? Like, why would some species want to be monogamous and not others?
Sure. So that's a great question. And I think it's maybe useful to kind of divide it, at least when we think about mammals, sort of rodents, or I would say primates, and sort of what we call below primates, and then primates and above. And, canids and dogs, they're maybe a separate entity. For most species, the largest driving force seems to be sort of female distribution as a consequence of resource scarcity. So if you take sort of I think the most recent estimates are, at least in mammals, maybe 60 times that monogamy has evolved. And if you look at that pattern, you'd seem to have a species that gets distributed over a large territory where resources aren't necessarily abundant, either seasonally or, sort of just geographically. So then, if you're talking about a promiscuous species, the females are sort of left with, raising their young and then having to sort of defend against intruders, defending against, predators, things like that. And so in that context, there is sort of this evolutionary question that, is influenced by the female, certainly in terms of her wanting to keep a male around, but then the male deciding, is it better for me to risk my offspring with this female being killed or dying versus defending them and co-parenting? Because that's also a really important feature that sort of co-evolved, is that in almost every, at least mammalian species, you see very strong co-parenting behavior, even though they are independent. And so that sort of evolutionary toss-up seems to be one of the major motivating factors for most mammals, that it's in the male's best interest genetically, as well as sort of resource provision wise, to defend his offspring and his mate against other potential mates. And
I see. So if we think about this in terms of like David Attenborough nature documentaries, you see something like the mother bear, and she raises the cubs by herself. And I guess basically what you're saying is the reason for that is the bears, as one example, live in environments where the mother is able to get more or less everything she needs food-wise, et cetera, for the cubs. And there's really no need for having that second parent there. But other species, I guess humans would be an example. If we live in more difficult environments where food is more scarce, you need to have, both parents either partitioning what they're doing to help rear the young, and have one or both of them being sort of the ones going out into this sort of more sparse environment and bringing things back so that you have everything you need for the offspring.
Yeah. So, and I think that actually may be part of why there's this distinction between sort of primates and below and primates above, is that with primates that are like altricial, right? The young are so helpless when they're born. You really need a lot of resources to provide for them for their survival. There's also another interesting sort of potential correlate, which is that in many sort of primate species, or even, if we again go back to the canids, you also have multi-generational sort of sets of animals living together. And so there's a much more complicated, in some cases, I don't want to say that this is something that defines the rule. So there's a lot more complex social interactions that are mediating sort of who is mating with whom, who can mate with whom, but more importantly, who lives with whom and who protects and shares resources. And so you get a much more complex structure where there is, at least modeling wise, a benefit to sort of having a single mating unit that then sort of creates an intergenerational sort of kinship or family unit. And then depending upon how animals leave that eventually, that sort of helps facilitate them forming their own clan, one, with its own numbers and its own sort of ability to provide. And again, there's a little bit of a debate between how that really influences and whether that's cause or consequence. And I should also just add that another factor that comes in, at least to the models for primates, is this idea of infanticide, right? That where you, and certain people sort of have data that support it. Other, there's contention that where you have species that may also demonstrate infanticide as a consequence potentially of either genetics or resource scarcity, that actually then also facilitates that even more, the need for sort of biparental care to protect the offspring and sort of,
I see. So, I mean, again, using the David Attenborough method of coming up with examples here, I've seen many nature documentaries where you have a pride of lions. Right, there's sort of the dominant male, and he effectively has a harem of females. Eventually, he gets old and feeble enough, and a younger male will come and sort of take over the pride. And one of the dark aspects for that species is very often my understanding is that the new male will basically murder all of the infants.
Right. And interestingly, in cats, there isn't actually a high incidence of monogamy. So those are some of the data that maybe call into question sort of the primary role of this like sort of infanticidal drive. And I think it's a question up for debate. It's also, it's a phylogenetic question that I think only more and more data really can sort of help us model better.
Yeah. Is the extent to which a species is monogamous also influencing the extent to which they are sexually dimorphic, generally speaking? Because I can imagine that if you have, let's say humans, for example, and other primates, a truly helpless young baby that needs constant care, you really have to have at least one parent who's always going to be there. And then because they always need to be there, they might have different sort of attachment styles, different modes of defensive aggression. And the other parent might have to be more exploratory, have to be more risk-taking and sort of go out into the world and do different things. Is there a relationship there?
So there is, and actually, So you're bringing up, I think, a really cool part, which is the behavioral dimorphism. But then another interesting thing that many species, whether we're talking about mammals or not, are very sexually dimorphic anatomically, right? Like obviously the peacock being one of the best examples, right? That and there's all sorts of sexual selection theory that is sort of models why you see such blatant differentiation in terms of sexual competition. But one of the interesting features, both anatomically as well as behaviorally, is that when species have evolved to become monogamous, they actually become less dimorphic anatomically. And then, as you were alluding to, for example, from the context of parental behavior, that includes behaviorally. And so one of the models that might come up with is that you're actually looking at sort of a de-differentiation from a sex different standpoint of species as they evolve to become monogamous. And one of the things that actually our work is actually showing is that that doesn't seem to be the case, because when you actually start to look at or manipulate the factors that are facilitating monogamy, you actually see sort of the behaviors almost sort of collapse back to more dimorphic behaviors. So that the brain at least has evolved on top of that sort of original dimorphism to converge onto behavioral circuits, for example, for parental behavior, that even though paternal behavior and maternal behavior, obviously in the absence of, with the exception of nursing, look the same, they're actually coming at it from slightly different strategies, at least genetically and presumably neurally as well.
Interesting. And I guess that would fit with what I know to be true for humans, which is that if compared to other great apes, we tend to be more monogamous, and we're actually, even though we are sexually dimorphic anatomically and behaviorally, we are less so than gorillas and chimps and so forth.
Exactly. Yeah, and that's true even for rodents, right? And if you look at them in the wild, that it's much more. I mean, for us, it's obviously even more difficult to tell sometimes, which is which. But that tends to be the sort of the pattern across phylogeny.
And so, we're thinking of all these interesting questions. A lot of them have to do with primates and humans. People are, naturally interested in humans because that's what we are. But of course, you can't really experimentally study humans in very much detail. It's very difficult and time consuming to do this. But there are other species that happen to be the ones that you work on where some of these questions can be tackled in a more experimentally rigorous way. So can you talk to us a little bit about the prairie voles, maybe starting with their natural history and just what we know about their behavior and some of the similarities between them and us?
Sure. So from an ecological standpoint, sort of like in the 60s and even a little bit before then, people were very interested in sort of just patterns of animal behavior and animal survival, right? And the original studies were sort of in the middle of the United States, southern Illinois, looking at just the behavior of these animals, trapping them. What is their seasonal variation? How does it vary with regards to rain, resources, things like that? And it was actually studies from Lowell Getz, who originally was trapping these animals. When I think he was a postdoc or grad student at that time, or postdoc, and noticed that in certain parts of the fields, like for certain areas of these sort of fields in southern Illinois, instead of trapping like one animal at a time or like a couple of animals at a time, he would often trap two animals at a time, and then they'd be tagging them. And then if you were then he noticed that if you were trapping the same animal, you were likely to co-trap the other same animal again. And then they started to look at these pairs, and it turned out that very frequently, with a much higher frequency than other areas or other species, you would see males and females together. And so that led to this original sort of potential hypothesis that these were animals that, for at least whatever purpose, were sticking together as male-female pairs, which sort of tends to go along with monogamy. And so that was sort of the original observation that led to this being identified as a monogamous rodent species. And then very soon thereafter, thinking about the sort of now underlying endocrine and neural mechanisms, a researcher named Sue Carter sort of postulated, based off of, and she's actually in interviews even said, based off her experience as a mother, one of the most profound sort of hormones that we knew was implicated in maternal bonding was this hormone oxytocin. And that's sort of what gave rise to this entire field of thinking about what is the neuroendocrine regulation of what ended up being monogamous mating strategies. But that's sort of where it first came from, these original ecological observations, long before even we knew that there were hormonal players, let alone what they might be.
And so the prairie voles, like how do they differ from just sort of the everyday rats and mice that people might see running around outside? Are they living in big social groups underground? Are they sticking together in pairs? What is their social
So
behavior? Yeah.
yeah, so I think voles are actually an interesting subset of rodents. That one of the questions is why they may be so convenient from an evolutionary standpoint, that have evolved many different, like in terms of monogamy, multiple times across actually multiple geographic regions, both in the United States as well as other parts of the world. And so they, as the name suggests, prairie voles come from the prairies, where you have, again, this large distribution with resource scarcity. There are closely related montane voles. There are mountain voles, meadow voles, which are slightly different ecological territories, and they've been named. They're bank voles, and different types of geographic names for some of the species that come from Europe as well. And so one of the features seems to be that they are very good at rapidly differentiating, both physiologically and behaviorally, into different ecological niches. And so that's been, from an evolutionary standpoint, an incredibly useful set of comparative studies, independent even of when we're thinking about behavior. And as I mentioned, so they're basically in the same class of rats and mice, but they tend to, depending upon their sort of social structure, prairie voles tend to have a den with at least the litter, and usually you have sort of an adolescent generation that often is sometimes sticking around, sometimes slightly older. They have what's called alloparental care, so they'll actually care for offspring that aren't necessarily their own biologically. There's there are
Yeah.
probably genetic as well as behavioral components based off of who you're living with.
Mhm.
That
It's like
also
the older sibling, making you lunch when your parents are at work or something. Yeah.
exactly. And so for prairie voles, that's sort of the context that they live in. Other vole species have, sort of mouse-like social structures. Rats tend to be hyper social, even though they don't necessarily have pair bonding. And so, depending upon the sort of ecology of where these have evolved to live, they have, varied social structures.
Mhm. So prairie voles out in the wild, how does the female meet the male? How do they start to form a pair bond? And walk us through what happens, through the courtship phase, through the offspring phase.
Sure. So, I think there's a little bit of looking for a mate, right? So, if you're a female, both males and females tend to have some sort of territorial coverage. Males often are the ones that will sort of seek out different territories, in particular if they're wandering away from the nest. And when they encounter a female, one other interesting evolutionary correlate that I think we haven't and others haven't looked at in depth in the wild is something called incest avoidance. And so monogamous species also tend not to mate with siblings. And I mean, that's a direction we can go in, because we've done some work to figure out, is that genetic or is it maybe not? But even in the wild, there tends to be this likelihood of not mating with a sibling. And so, if both animals are what we call sexually naive, so virgins, then it's really sort of the initial mating that seems to facilitate both the molecular and the neural mechanisms that cause this pair bond to form. It happens on slightly different timescales, at least when we look at it in the lab. Females tend to be able to form a partner preference, at least very rapidly, within 6 hours of encountering a male. Males tend to take a little bit longer, depending upon the situation of the study, maybe a day or two. And one of the things that seems to be at least important is sort of the interaction surrounding mating. And that doesn't necessarily mean like sort of successful mating and pregnancy, but rather the sort of mating attempts that stimulate the induction of hormones associated with mating. So for females, it's estrus. These are not cycling animals, they're induced into estrus. And for males, it's presumably some of the hormones that are activated in the context of mating attempts. Potentially being one of them.
So again, things happen on different time scales, but some of this can be quite rapid. And I would presume for this species, a lot of this is mediated through olfaction. They're smelling something on the potential partner, and whether it's a hormone or a pheromone or something that can trigger some of these attachment mechanisms to get started.
So I think that may play a little bit more of a role in the identification side of things. We know, at least for females, that actually like mating attempts stimulate the induction of some of these hormones. The pheromones are part of this whole equation. They're there. We know that they definitely play a role in priming the animals to be more sexually active, which probably, like you were saying, activates the circuits. And maybe even in a maybe with some specificity, but also, certainly with the level of more general activation for mating. And then as the animals spend more time with each other, investigate each other, and pheromones in particulars, right? So most mammals, other than humans, use sort of two different types of smell. They use olfaction, which is what we are used to, smelling things in the air, but then there's also pheromones, which are not necessarily volatile. And so they're the direct interactions with, whether it's tears or whether it's, at the animal's bottom, there are pheromones that are specific to these regions that are male and female specific that also probably mediate, at least in large part, how they identify individuals. And so now we're getting into the sort of the neurobiology of attachment, which is how do I know that this male or this female is different from basically every other male or female? And that's the one that I'm going to attach to. And some of the work that we're actually doing is suggesting that it may not only be olfaction or chemosensation, that there might be other sensory modalities that they're using, and maybe even one sex versus the other, to identify who's my mate versus who's not.
Interesting. So I was at the dog park yesterday, and everybody knows when you get a group of dogs together, they walk around and sniff each other's butts. But what they're actually doing there, to sort of connect this to what you're saying, is my understanding is that there's actually glands that are secreting pheromones and other things, and they need to get right there because, as you said, a lot of these things aren't volatile. So they're basically identifying individuals by physically putting their nose against things that their nose can pick up.
Right, and these compounds, right, are actually aqueous, and they, how they're even perceived at a neural level is via sort of an aqueous medium in a different part of, this general chemosensory pathway. Just in the same way, like our nose picks up volatiles, these things pick up compounds that are generally in aqueous sort of environments. So that I think the dog analogy is exactly illustrative of what we're talking about.
And you mentioned there's sort of olfaction per se, and then there's this other nose-based sensory apparatus that has to do with pheromones. Everyone has experience with dogs. We know that some species, like mice, seem to they use their sense of smell a lot. And I know that this gets complicated and a little dicey, but in general, people characterize primates as being highly visual, and that maybe we don't use our nose, at least in quite the way that some other species do. My question is really just, before we get into the oxytocin stuff, do humans actually sense, do humans have pheromones? Do we have that mode of perception?
So the safest answer, I think, and the answer that I think is probably true, is I think the jury's still out. There are a lot of data that suggest that we do sense sort of chemosensory compounds that are related to our biology. The question is to whether or not they're volatile versus non-volatile. So you're probably aware of, and there are these studies that are associated with like scent of sweat, right? And that how that modulates attraction, and that also, at least in some of the early studies, changes for women's with their cycle. And for men, it varies in terms of like health and things like that, how they produce some of these compounds. And so whether that response is actually coming through what is this sort of accessory olfactory system, or whether it's sort of a modification that humans have to their primary olfactory system, I think is certainly out. We know that there are these sort of, they're not anecdotal. I mean, these are studies that were done in large numbers of individuals, but I think from a sort of neurobiology system standpoint, the jury's still out in terms of whether we genuinely have what we call pheromones in most other species. And, other primates, non-human primates, definitely seem to have, or subsets of them definitely seem to have preservation of the system that we're talking about. And so I think it's still a question of how much do we rely on those versus are they potentially there? Maybe they influence us in ways that, we haven't really teased out for a lot of the reasons that you were alluding to in terms of, well, you can't do these experiments in humans. But in general, primates tend to be more visual. And, I think that potentially is the analogy in terms of how we think about the sensory systems involved in identifying a mate versus, somebody else.
Okay, so going back to the prairie voles, boy meets girl, they hit it off, and they start mating, and then the female gets pregnant. So after she gets pregnant, how do the behaviors of the male and female start to change?
So it's actually even before they get pregnant and even independent of pregnancy. And so there's an interesting component that, I think is a separate set of biology associated with pregnancy, in particular, what happens in the male brain following, either the hormone signals when the female is pregnant or even after delivery of pups. And there's an interesting, we were talking about infanticide. So there is this, in rodents at least, what's called the infanticide clock, that if a male mates and then there are pups around about 3 weeks after, he's not infanticidal towards them because the likelihood is that those are his pups. And so we know that there are hormone signals that sort of are communicated or perceived by the male surrounding pregnancy and mating. But for bonding, that doesn't seem to be a requirement. So within those first few days, independent of pregnancy, whatever it is that happens from a neuroendocrine and neural perspective, between males and females that are induced during these initial social and mating interactions, triggers two things. And those are sort of the principal components of what we call sort of, from the social behavior standpoint, pair bonding between the two animals. One is that the animals develop a preference for each other over now other animals of the opposite sex. So when you actually test them and give them a choice, they go to their intended, or their now, however we want to call it, betrothed or bonded partner, in contrast to spending time with the stranger animal. The other part of this, though, is this flip that I was talking about in terms of the rejection behavior. It's not just that they actually prefer. When confronted, for example, with just a novel individual, they actually will actively reject these animals. So it's basically a sort of display, and I don't want to use the term aggression because I think aggression is different than this sort of rejection. Mm-hmm. But there's a lot of behavioral overlap when you just sort of look at the behaviors. And one of the questions is there neurally overlap between aggression versus this rejection behavior? But you can imagine from an evolutionary standpoint, you're kind of going against basically 4 billion years of evolution that says mate, 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 to say, okay, even though you have almost all of the same signals as my partner, I'm actually going to actively reject you. And so those are the sort of two predominant components of what pair bonding really results in and manifests as.
Interesting. And then so then when they have offspring, what's going on, and what's going on behaviorally, and what's going on underneath the hood that is underlying the sort of attachment phase of the parent with the babies? So
behaviorally, both parents will actually demonstrate really, really robust parental behavior, right? I mean, the one exception that's different is nursing, right? But mm-hmm. both animals actually will crouch over the pups in the nest, providing warmth, providing protection. Both of them will actually groom them. Both of them will kind of attend to them. Both of them respond, for example, when a pup is separated from the nest, to actually retrieve it. And that's a response that's generally guided by the pups vocalizing. And so you really see sort of a significant parallel pattern of parental behavior between the maternal and the paternal behaviors. What's happening under the hood is a really good question. And that's actually one of the questions that we're interested in pursuing is how does, in the context of this pair bonding, how do we get the demonstration of these behaviors, in particular in males, right? Maternal behavior is sort of generally expressed across mammals, with varying levels of sort of fidelity in terms of when and how robust mothers display these behaviors. But paternal parental behavior is pretty much an exception, unless we're talking about biparental care. And so, as I was talking about whether or not this is coevolution onto kind of a common final pathway, or in some way, shape, or form, these circuits, which maybe existed in males but weren't necessarily active, have now been co-opted by these signals by mm-hmm. virtue of this bonding circuitry molecularly and neurally. That's an open question.
I see. So both parents take care of the young. They do the same types of behaviors. It could be that there's sort of a shared set of neural mechanisms underlying both, or it could be that sort of the male brain and the female brain have done it in different ways.
Right.
Interesting. So how long does it take for the young to grow up? And what are when do things like oxytocin come into the picture, and what exactly are they doing?
Oof, that's okay. Very big question. So I think the question of when oxytocin comes into play varies on what behaviors we're talking about. And so, we know from many, many, many different species that oxytocin is involved in parental and pup sort of attachment, in particular maternal attachment. But, certainly we would argue that it's probably also playing a very, very, very large role, and pharmacology supports that for paternal attachment as well. And so then, these animals basically are they grow up for 3 weeks, at which point, at least in the laboratory, we can then basically what we call wean them. So they're then basically self-sufficient. They can eat on their own. They, can sort of take care of themselves. They are social species. And so this is another interesting question, is that if you actually isolate them from siblings, there are different developmental consequences. And so that the period of adolescence, which kind of goes from about, 3 to, let's say, 4, 5, or maybe 6, 7 weeks, depending upon the situation, laboratory, or, sort of the context, is a period in which these animals actually form peer attachments that can be even between members of the same sex, or interestingly, actually between sort of siblings, whether they're actually genetic or non-genetic. So if you raise these animals together, they'll form attachments to each other, and that intersex attachment actually has a potential role for what we were talking about with terms of incest avoidance. That it may not matter whether you're genetically related, but if you grow up together, you become familiar and attached to each other, but then that sort of leads to you not necessarily wanting to mate with each other.
I see. So there's probably some kind of fingerprint, whether it's olfaction based or something, and it's not necessarily just to do with your genes. And there's something that's in group versus out group distinction, basically.
So, okay, and that's basically sort of the other part of all of this is within group, without group, and how is that communicated, right? But at least at the first approximation in the lab, it's not genetically dependent, and something that's coming from environmental, cues or sensory cues exchanged between the individuals. And so then after sort of, you have, again, this organization activation, the gonadal hormones sort of kick in, you have sort of the final sexual differentiation of the nervous system and behavior, and then these animals are now reproductively active, right? And so that is where now the sort of phenomenon restarts of bonding between individuals that mate with each other. They do preserve those sort of peer affiliations from that early adolescent period. So, animals will show a same-sex preference for animals that they're familiar with versus non-familiar with. But then in terms of mating, that's what sort of results in the pair bonding for the next generation. And the likelihood is actually, based off of pharmacology, that things like oxytocin and vasopressin are playing a role at each of these stages in different ways, and presumably on different subsets of these neural circuits.
Can you talk about, for people, like, what is oxytocin? What kind of molecule is it, and how does it work at the cellular level?
So it's a hormone, but unlike, for example, the steroid hormones that we were talking about, made by the gonads, it's a peptide hormone. So it's a 9-peptide hormone. It's very closely related to another 9-peptide hormone, vasopressin. They both seem to have diverged from a common precursor that's actually conserved, across phylogeny. Even like, worms have these precursors. And so these hormones are, they're made from sort of larger peptides, and then they're cut into smaller pieces in different neuroendocrine populations of neurons within in most cases, the hypothalamus. The one that we often talk about is something called the paraventricular nucleus. It's a principal source in most mammals of both of these hormones, as well as other peptide hormones as well, even though it's not the only one. And so these hormones are made, and they go in sort of two directions, literally. Part of it is into the periphery, which is secreted via the pituitary, and they govern physiology sort of throughout the body. They're involved in parturition, they're involved in various aspects of lactation, things like that. But then when we're talking about this in the context of pair bonding, they're actually secreted synaptically to regulate the activity of neurons sort of postsynaptically. And these hormones bind to what are called G protein coupled receptors, and they're sort of the largest family of receptors sort of in the genome. And by virtue of binding of these peptides to these receptors, there are intracellular processes, like molecular events that happen, enzymatic events that change the levels of certain molecules, that influence both how a cell, or a neuron in this case, can respond to other signals, as well as the baseline physiology of those cells.
Interesting. Okay, so these things are peptide hormones. So they're basically small proteins. And part of what they do, at least, is to basically act, at least in certain cases, like a neurotransmitter.
Right, exactly. And they fall into this category of neurotransmitters called neuromodulators. And so, right, they're often sort of coupled with some of the more direct excitation versus inhibition neurotransmitters to really sort of influence how the signaling is actually happening at a synaptic level.
And so I imagine that you guys and others have done experiments, with the prairie voles, probably with other species as well, where you're sort of adding more oxytocin at, some time when it's not supposed to be surging, or you're taking away at a time when it is. So how do some of these attachment and parental and other behaviors change in response to, adding more oxytocin or taking it away and things like this?
So that was sort of the foundational aspect of the field is, based on those original observations that Sue Carter made about the fact that it seems to play a role, people started to manipulate. The other part of this is actually people also started to compare, and that's a different direction that I'll talk about in a second. But when they started to manipulate oxytocin, either like you were saying, giving it in the context of social situations where it might not yet have been released or wasn't released, but for a shorter period of time, versus blocking it. And there are drugs that can do both of them, and fairly specific drugs that really either you can give exogenous oxytocin, which predominantly acts to the oxytocin receptor, or a drug that blocks it. It had what sort of the predicted effects based off of what we think these molecules are doing. When you gave oxytocin to animals that were put together for a shorter period of time, they developed a partner preference, very quickly relative to animals that got like just a vehicle. Similarly, when you gave either systemically or then by virtue of those other studies that I was talking about, local either oxytocin to facilitate or blockers to prevent it from working, you basically disrupted the formation of these pair bonds. So if you put animals together and you kept on giving them an oxytocin antagonist, you prevented pair bond formation. And interestingly, similar findings were found in slightly different contexts for vasopressin, this other related hormone that has its own receptor.
Mm-hmm. And so what happens when you take away oxytocin or the ability of the body to respond to it?
Depends who you ask in terms of the animal and the species. And so that was the experiment that we did is, if it's okay, I'm going to kind of take a little bit of a side note and talk about the receptor distribution and kind of why the receptor question is actually a really important one too.
Okay, perfect. So like where the receptor is in the brain.
Exactly. And so because, people were really hypothesizing that oxytocin and then vasopressin were playing a really big role in these behaviors, they looked to see, okay, are these genes different? Are the neurons that make them really different between a monogamous vole species like a prairie vole versus a promiscuous species, let's say a meadow vole or a montane vole? And when you look at the peptides, there are very subtle differences, but there really aren't major differences. But as you were speaking to, peptides act on receptors. And so by virtue of the hormones having very large regions where they project to, they then looked at where are the receptors for oxytocin or vasopressin in a monogamous versus a promiscuous species. And that's what sort of blew up in the field, was that if you look at a monogamous species and multiple monogamous rodent species, for example, you see that the oxytocin receptor or the vasopressin receptor are reproducibly in different parts of the brain in monogamous species than in promiscuous species. And some of those parts of the brain are ones that you can create pretty good models for their involvement in reinforcing different types of behaviors. So for example, the nucleus accumbens, which is involved in reward reinforcement, also involved in sort of aversive reinforcement as well, expresses or has a lot of oxytocin binding in a monogamous species.
So just to really simplify that, the way I guess a cartoon way of thinking about that would be, so nucleus accumbens, people usually think about dopamine, they think about drug addiction. People get addicted to their kids. Parents, it's as if you're addicted to your offspring. Is that, how you would start to think about why you might have an oxytocin receptor there?
Sure, and that's exactly how, and that's sort of the model that it's you have to reinforce these behaviors in order to make facilitate them for being beneficial to the animal, or for the animal to actually want to engage in them. I shouldn't say. I mean, the benefit is the evolutionary side of things, and all of the associated circuits, parts of the prefrontal cortex, right? Where do I keep the sort of initial or the long-term memory of my partner? Parts of the other parts of the limbic system that are involved both in mating, aggression, as well as just our response to sort of social stimuli. So there was this really, really profound difference in terms of where it appeared oxytocin and vasopressin were acting in a monogamous versus a promiscuous species. So that set the stage for all of the studies that really said, okay, can we start to, for example, initially, because prairie voles weren't mice and didn't have all of the genetic tools, can we start to pharmacologically manipulate oxytocin or vasopressin in these different subregions of the brain to really say, okay, how are these different regions or different parts of the circuitry contributing to aspects of this pair bonding behavior. And that's sort of what 30 years plus years of work has really sort of demonstrated that different regions are contributing in slightly different ways, but that by and large, when you, for example, inhibit oxytocin or vasopressin in some of these regions, you really significantly impair the animal's ability to pair bond. And so that led to the experiments that we have recently, or not recently, we've been working on this for quite some time, that we did, which was to bring molecular genetics to the prairie vole. And part of that is kind of motivated by my own sort of way of thinking about innate behavior. And that's, I come from this molecular genetic perspective, thinking about using the genetics of a species to really identify the neurons that are involved in innate social behaviors, right? And so, again, because the receptor is in these different places, it points you to the different parts of the brain that might be involved in social attachment.
Mm-hmm.
But prairie voles weren't mice. They didn't have the decades of molecular genetics and cellular biology and embryology that sort of has allowed us to really develop tons and tons of tools in mice and sort of other quote unquote model organisms. There were species that really was, had a rich behavioral history, rich pharmacologic history, but we didn't have the ability to genetically manipulate them. So we developed those tools using CRISPR-Cas9 eventually, and there was a precursor to that of about 5 or 6 years that we can or don't need to go into. But we used CRISPR technology to, in this case, knock out the oxytocin receptor for 2 reasons. One was just as a proof of principle that we could genetically manipulate things in a prairie vole and then really start to do sort of the next level of molecular genetics and neurobiology in the species to really get at the circuits and genes involved in pair bonding. And then also, to sort of just ask the question, which we expected a very specific answer, that, what is the genetic requirement for oxytocin receptor signaling for pair bonding? With the idea being that it would have a pretty significant impact on the ability of these animals to form.
I see. So before we get to the result there, so every expectation I think people would have is, we know oxytocin is important for some of these things. We know that the distribution of receptors in different parts of the brain is different between monogamous animals and non-monogamous animals. Obviously, you would imagine that if you get rid of the oxytocin receptor, something major is going to happen. Before you describe that result, so you used CRISPR to get rid of this receptor in prairie voles. Did you just knock it out completely everywhere, or did you do it for certain brain regions, or both?
So the first experiment that we did was to knock it out everywhere, and so we made what's called a knockout animal, and that again, from the proof of principle standpoint, is the sort of the principal technology that allows us to develop all of these tools in mice from a transgenic perspective, right? So you can use things like viruses to manipulate genes in certain ways, but at the end of the day, you need to make different forms of different genes, different alleles, right? Either to conditionally manipulate them, like you were saying, in different parts of the brain, or to build tools to label neurons by virtue of the genes that they express. And that's sort of what's led to the revolution in molecular and circuit neuroscience in mice, is because we can define circuits based off of the genes that they express, define populations of neurons based off of the genes they express, and then manipulate them using the sort of intersection of these two types of technologies. And so what we did was to really spend a lot of time being able to bring CRISPR into the vole embryo. And that involved just a lot of time understanding how vole embryos develop, what's the biology, at the other end of the animal instead of the brain, that really allows you to manipulate them at a genomic or, sort of genetic level.
Mm-hmm. So eventually you guys figure it out. You get the technology to work. You knock out the oxytocin receptor. What happens?
So we were surprised that not as much happened as we initially expected. So, I often talk about now that my postdoc, who was doing a lot of the studies, we made the animals. Initially, there weren't any obvious phenotypes, like they didn't have severe growth deficits or anything like that, which I don't know that you would necessarily expect with loss of oxytocin receptor. But then we sort of, what would say, cleaned them up genetically, and then started to look at their behavior, and we're actually quite surprised that when we looked at the behavior of animals that had been put together for a significant period of time, they showed very, very robust partner preference. And then in collaboration with sort of actually the lab the mentor that I did my postdoc with, and our collaborator, so Nirasha and Karen Bales, we looked at multiple alleles, and we started to look at different aspects of their pair bonding and parental behavior, and found that they do develop very, very robust partner preference and demonstrate very robust parental behavior, which, I think based off of the pharmacology, I would say was a little bit of a surprise, because there was really this sort of idea that, from the perspective of disrupting oxytocin signaling or facilitating it acutely, you really had profound changes in the ability of these animals, or the propensity of these animals, to form pair bonds. But then at a genetic level, clearly that wasn't the case. I will say that we are looking at other aspects of the behavior, and perhaps not surprisingly, as a sort of a neurogeneticist, we see that the genetic architecture of pair bonding behavior actually is, it's multifaceted, right? And so whereas this initial disruption wasn't something that's prominent, there are discrete modules that do seem to be dependent upon oxytocin receptor signaling. What isn't the case is the ability to form a preference for your partner.
Okay, so a lot of these behaviors are intact, even in oxytocin. If you get rid of the oxytocin receptor everywhere using this CRISPR technology, but you just said that there were some things that were disrupted. What were those?
So the sort of two areas where we're really focusing is on the reciprocal behaviors between these animals, right? So how do animals actually interact initially between each other? Eventually, they clearly can form a partner preference, but are there actually subtle differences in sort of the social interactions that they engage in? If one of the animals is lacking the oxytocin receptor and the other one isn't, like, can they detect it? Do they behave differently with one versus the other? How is that happening? And so there is definitely this sort of overall effect in terms of probably the patterns of social interactions that facilitate pair bonding are altered, even though the animals eventually can form this partner preference.
And so, yeah, it's a surprising result. Sort of the naive expectation I think most people would have had is, okay, if we get rid of the oxytocin receptor, we're sort of going to break all of these behaviors that we've been talking about. But you generally don't see that. And where my mind goes with this, I started my training in evolutionary developmental biology, and one of the sort of core things that I learned from that era of my life was the relationship between redundancy and robustness in development. So a lot of times, you'll have multiple copies of a gene or a protein or something, and these things are highly overlapping in terms of what they're doing and where they are. And the basic idea is, development is complicated. It needs to go right, and there's lots of ways and places it can break. But if you've got multiple redundant versions of things, if one of them breaks, you've got a backup, basically. And so you mentioned earlier that there's this other thing called vasopressin. It's also this sort of peptide that's very similar. Do you think something maybe is going on here where you've got redundant peptides that can compensate when one is gone?
So there's sort of two questions there. And one is sort of the redundancy from the peptide perspective, but then this is actually also talking about the receptor. So then, we'd sort of be making the argument that vasopressin receptor may now be sort of playing the role for where the oxytocin receptor was. We didn't see that. When we looked for sort of significant changes in the vasopressin receptor expression in the oxytocin receptor mutants, we didn't see that. There may be, in very, very specific subregions, a place where vasopressin upregulation or receptor upregulation is, or even where it is generally, is somehow responding to oxytocin. But at a global level, that's not where we're seeing the redundancy. I think what probably, apropos of your point from an evolutionary perspective is, these are behaviors that are absolutely critical to the species, right? And so there is a fundamental difference between disrupting a neuromodulator in an otherwise wild-type animal that's developed with all of the neural and developmental homeostasis that allows it to be a functioning wild-type animal, and then you come in and you dramatically alter the function of these circuits, versus, as you were suggesting, when you don't have the gene itself from the get-go, there are redundant or compensatory mechanisms. And that's an open question, is one of the things we're looking at is what are the changes in gene expression that happen plus or minus the oxytocin receptor that compensate for the absence of oxytocin receptor signaling. And, again, I say that we were surprised, but, I think if we take a step back from an evolutionary perspective, you're not going to have a single point of failure, right? These animals have to have robust systems, or evolution would suggest that they need robust systems to really make sure that they can maintain themselves and propagate, right? And so it's not going to be that suddenly if a single signaling pathway is slightly disrupted, or in this case globally disrupted, that they can't still facilitate their reproduction.
I assume that this is a direction things will go as the technology develops and gets more and more precise, but just in sort of molecular developmental genetics, broadly speaking, you often see differences in experiments where, you do a whole gene knockout right from the beginning, and that sort of gives space, for, so to speak, to the body to come up with as much compensation as possible, versus, you start to develop ways to knock out the gene in one place at like a very well-defined window of development. Do you think that's where things are going to go, and that you'll probably sort of unmask some more significant phenotypes when you do those more kind of spatiotemporally targeted?
Yes. So I think the short answer is yes. And that's certainly the direction that we would like to go in, is to be able to manipulate the neuron, or the receptor expression, after an animal has had the chance to develop, and see what are the effects, sort of with local distribution or even developmental. I mean, you could do it both ways, right? And, to your point about the more sort of both subtle phenotypes, but also more maybe profound effects, is, there are other aspects, like I mentioned, this, the flip in rejection behavior, right? That seem to have more of the oxytocin receptor signaling seems to have more of a role there. And so I think both in terms of what the global knockout is really showing us is that there is this sort of circuit/genetic separation in terms of how these aspects of attachment behavior are being regulated. But I think the question of what happens when we just take it out in the context of an adult animal is a really, really important one. And also is these are all orthogonal in terms of really understanding what are these circuits doing, what are they responding to, and then both at a sort of global circuit level but also at a synaptic level, what is oxytocin receptor signaling doing in different subsets? Because even though the receptor is the same, there's only one receptor in the genome. It's going to have different effects depending upon the cell type that it's in, whether both molecularly within the cell, but also it's the same receptor that's doing the same thing, but it's going to have different consequences, but also different cell types. Like if you have it in an inhibitory neuron versus an excitatory neuron versus a serotonergic neuron, you can imagine all of these things will have very, very different readouts for behavior.
So going back to behavior and the natural ecology of the prairie voles, and trying to tie a couple things together. So we talked about earlier how sort of one theoretical reason to explain like why monogamy evolves in certain lineages has to do with resource scarcity, basically. And we've talked a little bit about, we've talked about pair bonding, in-group versus out-group distinction, incest avoidance, things like that. So like with all of that stuff in mind, I can imagine that, okay, you've got a pair of prairie voles, they've got a litter, they raise the litter, and it becomes time for the kids to venture off into the world on their own. And on the one hand, you want to take care of your offspring. They're your offspring, of course. On the other hand, if you're a prairie vole and you've specifically evolved this reproductive strategy because of resource scarcity, at some point you've got too many mouths to feed, and maybe you need to kick the little ones out of the nest. In fact, I used to have pet parakeets, and one of the behaviors I observed when I enabled them to have a nest and mate is, the mother they would both, there was biparental care. The mother would stay in the nest the whole time, but at some point the babies got big enough and loud enough and hungry enough that she basically became aggressive, and she would attack them until they left. Do you see that type of thing with prairie voles? Do they sort of actively make their young go away?
So in the wild, there definitely seems to be something that facilitates the sort of the older young from leaving the nest. There hasn't been very, very reproducible documentation of aggression per se. And it may actually coincide with the young leaving when there is larger resource availability to then go find their own territory. But to what you're talking about, that is actually something that's seen, for example, in dog canine species, is that you actually have, at some point in time, you have reproductive suppression by the mating pair, so that the subordinates don't mate until, like you said, the nest gets too big, and then they're ones that are kind of have a higher likelihood of mating that then they force out for exactly that purpose. That also allows those animals to then go start their own sort of nest and clan. And so with prairie voles in the laboratory, we don't really encounter that to the certain extent because there's not huge resource scarcity, and we don't try to use that as a manipulation, but we also remove the young from the nest at some point in time, unless we're specifically looking for this alloparental care. In the wild, I would not be surprised if there is some level of sort of behavioral modulation of how they sort of deal with that in the context of either too many pups in the nest or when resources become scarce. But it's not something that we actively see. And in part, that's also because they demonstrate such robust alloparental care, right? So in many cases, you actually see that they're sort of taking care of the family, even though they're not necessarily reproductively active.
I see. And, one of the things that characterizes a lot of species is, when the mother has offspring, she obviously becomes very attached to the offspring and cares for them. But simultaneously, mothers will often become hyper aggressive or defensive, at least, towards intruders, right? They don't want, the classic example is like, my family's from Michigan, and my parents live in northern Michigan. So everyone knows about bears up there. And basically what everyone understands is, black bears are mostly not a threat most of the time. They're not going to bother you. They'll typically avoid you unless they're starving. The major exception being if the mother's with her cubs, in which case you just need to get the hell out of there.
Right.
So
akin to the elephant example that you brought up. Yeah, exactly.
Yeah. So thinking about oxytocin, so with that in mind, oxytocin, the sort of cartoon version that you see, out in magazines and stuff, is it's the love hormone, it's the cuddle hormone. It's just sort of the signal that makes you love whoever's around you. But I also know that there's some work out there, and I'm not super familiar with the details, that it might have a role to play specifically in the sort of in-group, out-group distinction thing, where it's sort of, if you're in the in-group, you're going to be more altruistic and display care behaviors and things like this. But if you're in the out group, people or animals might be more aggressive or more selective and things like this. Is that true? And what do we know there?
So that's certainly true. And, I think the example is, I think the closest example is maternal aggression, right? And the fact that, for example, the mother will defend against any intruder except for her partner, right? In terms of or I shouldn't say any intruder that isn't somebody that she is familiar with, right? So other offspring that are part of the clan are not somebody that she'll fight against, but a mother will very, very actively defend against any potential intruder, male or female, into the territory. And the same thing seems to be true, I would say, with some of the studies that have been done in humans in terms of in-group, out-group. There have been some studies looking at, for example, people who are in relationships who, if given oxytocin, actually show a stronger aversion to an attractive member of the opposite sex, because it facilitates what you're saying, this sort of othering. And, I don't, that's a term that has a lot of weight in different contexts, both in sort of the psychology literature, the sociology literature. So I want to be very careful about using that. But I think that what we're seeing is exactly what you're talking about, is that you're amplifying the response to familiarity from different contexts, whether it's your offspring, whether it's your mate, it's your siblings. But as a consequence of that, you're also filtering with a higher signal to noise ratio anything that's not familiar, right? So anything that is the other, which becomes very important for example, your response to a novel mate, right? How do I identify my mate versus another mate? They're going to be subtle differences. For us, we think everybody's face is so unique. I mean, they are, but probably to a vole, it's a primate that looks basically the same. Similarly, for us, there are going to be subtle differences between the vole's pheromonal profile, but those need to be amplified for an animal to identify, obviously with other behavioral contexts and cues, this is my mate versus this is not my mate. And if it's not my mate, then I actually evoke a very, very different behavioral response, which is this rejection behavior. So I think that is exactly the neurobiology that operates at multiple levels, depending upon how complex the social group is.
So they've done experiments in humans where you basically give exogenous oxytocin and people become more averse, or at least less attracted to attractive members of the opposite sex.
And I will say there's the one caveat that, sort of the intranasal oxytocin is still its own sort of debate in terms of how much of it actually gets there, but it did have an effect. So, I don't want to speak to whether or not the biology of what happens when you give intranasal oxytocin, but that has been an observation in humans. Yeah.
Interesting. Wow. So
sorry,
what? Go ahead.
You just reminded me of another thing. The other place where people have sort of looked at oxytocin is, for example, in people who are really, really into certain sports teams, right? And insofar as you can actually monitor in that context, your team scores a goal, people's oxytocin levels actually do go up, and right. I mean, that's been something that's reproduced, obviously in sort of smaller clinical situations, but seems to be the case, and also makes you, the higher the level of oxytocin you have, the more likely you are to then demonstrate some sort of aversion towards the other team, right? And so this idea that it's amplifying responses of who I'm with versus who I might not be with is pretty consistent.
I see. Yeah, and I'll let everyone ponder their own examples from their own life of the sports and in-group versus out-group behaviors. But it's a really fun area to think about. I want to ask you about, I guess we'll stick with the prairie voles unless we should go somewhere else for this, but what happens when you disrupt one way or another some of these parent offspring interactions, and the offspring are not getting the kind of parental care that they're supposed to be getting in a normal ecological circumstance. What happens? What does that do to the subsequent social and sexual behavior of the offspring when there's some kind of deficit in parental care?
So, and I think you already alluded to that. This is something you can start with sort of independent of prairie voles, right? So there's been great studies from Michael Meany beginning, and that was actually looking at innate differences in how rat moms sort of took care of their pups, and showed that pups who were the offspring of high grooming versus low grooming, being sort of like the sort of extreme measures of those initial groups, actually had reproducible patterns of parental behavior themselves. This is in particular for females. And so that, female pups from high grooming mothers grew up to be sort of, from the rat literature, good parents. They showed very, very attentive parental behavior, good pup retrieval, and a lot of grooming behavior, versus female parents female pups who were from low grooming moms grew up to be low groomers, and also, sort of that persisted. They also had other parental behaviors that showed a deficit. And so there's certainly a reproducibility and sort of a transmission of these behavioral consequences by virtue of these early life experiences. Similarly, in the context of both parental behavior and now kind of moving into prairie voles, parental deprivation has pretty significant effects and reproducible effects on late life patterns, both of just general social behaviors as well as pair bonding and parental behaviors. One of the things that we're also very interested in is, parental deprivation is incredibly severe, right? And we know from clinical populations, even in the absence of sort of the nutritional deprivation that can also be, a consequence of isolation. But if you take that away, for example, the Romanian orphanages studies, right, where you had individuals where they had all of the nutrition provided, but didn't have the socialization. In some cases, when we're talking about the kids that were there from the earliest stages of life, they developed what those clinicians called pseudo-autism, a very, very, very profound set of disruptions in their social cognition and social behavior, in addition to sort of other clinical sequelae. The same thing is also true in animal models. When you talk about this parental deprivation, it's a very profound effect. So then the question is, can we start to separate out the stages of social deprivation and also the specific effects of social isolation or social deprivation from other types of stress, right? Like thermal and things like that. And so that's actually something that, we and others are starting to look at in prairie voles, and are finding that there are these very reproducible effects, that if animals are sort of socially isolated at even later stages of their life, but, prior to adulthood, it does affect their propensity to form pair bonds. It also affects their propensity to, again, have this rejection behavior. So sometimes they become, in fact, hypersocial, and will form a preference if given a choice, but then also if just confronted with a new individual, they're totally willing to mate with that individual. And there's also interesting differences between the sexes for how these things play out.
Yeah, can you go into a little bit more detail there and maybe talk about an example or two?
Sure. So for example, if we take animals that animals that are socially deprived early in adulthood, not necessarily complete parental deprivation, but periods of social isolation.
Do you just mean sort of you're not allowing the parents to be there for some fraction of the time?
Right. So you can do that, like sort of individual or periods of isolation, or sometimes what people have done is very, very rigorously tried to say, okay, we're going to give them warmth, we're going to give them nutrition, but we're going to take away the social context, right? The actual parent, which again, how you measure the severity of the stress, I think is, that's its own question. And if you look at what happens to those animals, in some cases, what you see is that, for example, the females will form a pair bond, if again, given a choice, even though it's, the strength of that pair bond may be, depending on metrics, less severe. But then if you give them a novel male without their partner, they actually treat that as they would a novel animal, like not having been bonded. So
they don't reject him.
Exactly, exactly. And in some contexts, you see almost the opposite in males, that they sometimes become very asocial, or actually, depending upon the model or how you're testing it, they may be like more anxious. So they're not more pro-social, even in terms of the initial social interactions that lead to pair bonding. So they take longer. They have a less sort of robust pair bond. And then, depending upon the context and the level of stress, sometimes you see the similar phenotype to females where they become hypersocial. Sometimes you actually see that they're even more rejecting. That so they have sort of this hyper attached, even though it's less social phenotype.
I see. So in general, it seems to be that whether you're male or female, and the exact pattern will differ between the sexes. Not having the parent there as much, being deprived of parental interaction, seems to push them to one end or the other of the distribution of normal prairie vole social behavior.
Right, right. And that's consistent with those studies in rats where, when you had the females that were sort of raised by the low grooming moms, they not only were not necessarily the best moms, but they had much more frequent mating. So they would abandon their pups at an earlier time point in order to mate, and then mate and mate and mate.
Interesting. There's a couple directions we could go with that. I mean, one is a little bit more theoretical. I know that there's some ideas out there on basically you take the concept, which a lot of people learn about in grade school, of R versus K selection. So like, so you have some species like humans, for those listening, where we're sort of one extreme example where, we put a lot of time and effort into our offspring, so you have to really, babies are helpless. You have to be there and invest years and years and years of resource allocation and attention and care to them. Whereas you think about something like a salmon swimming in a river, there's none whatsoever, right? The females just lay eggs and the males just sort of, put their sperm in the water, and they try to have as many babies as possible, knowing that there's going to be no investment and most of them will die. Can we think about some of these behaviors within a single mammalian species like this, where one of some of these things that you're describing that we think of as deficits or something could also be thought of as different types of strategies for different types of environments with different levels of investment?
And I think that's a really, really important point to make, because, especially when we start to analogize some of these behaviors to humans, is, these things have evolved for many, many different selective pressures, right? And so the context in which what we call sort of a deficit in behavior may actually, there's sort of these evolutionary theories of various neuropsychiatric conditions and why certain things manifest in different contexts, which, that's its own conversation. But I think that what you're getting at is that, the plasticity of the circuitry is there, right? And in different contexts, you're going to have a different adaptive strategy for survival, like within a species. So there's kind of the intra-species versus the interspecies comparison that you were making, right? So we know that promiscuous species tend to have larger litters, right? Because of sort of that phenomenon that you're alluding to, that, it may be to their benefit just to put as many offspring out there as they can, hope that they survive, and, good. Versus a species where you have biparental care and you're investing inordinate amounts of resources into the offspring that you have. They actually have fewer offspring as a consequence of that. But in the context of either environmental or sort of social stresses, it may actually be very adaptive. For whatever reason, if, there's a couple more hawks in the area and your parents are, getting picked off, left and right, it may actually make more sense to sort of adapt a very different mating strategy in order to propagate your genome.
Yeah. And, your work is really interesting because you're studying these prairie voles, this very interesting rodent species, and doing this basic research, but you're also a psychiatrist. And, on the one hand, you don't want to anthropomorphize too much when you're talking about rodents. You don't want to overgeneralize the findings from one species and one slice of the experimental field that you're in. On the other hand, there are some really interesting parallels between the prairie voles and the humans, and it's really hard not to think about how some of this applies to many, many human circumstances that probably come up as people are listening to what you're talking about. How do you start to use your basic research findings to think about human neuropsychiatric conditions and social deficits and things like that?
So I think there's a couple of ways that I think about it. One is, certainly, actually, the very basic question is, what's the point of studying these behaviors? Even though we can sort of, anthropomorphize or make analogies at a behavioral level, is that really biologically useful, right? And that kind of goes to the point that, I mentioned that when we look at sort of monogamous rodent species, you see sort of convergence on oxytocin and vasopressin, but that seems to be across sort of phylogeny, that when species have evolved or just even sort of diverged different social strategies, these peptides come up more, often, if not every time. And then when we start to look at mammals, whether or not it's more oxytocin, more vasopressin, or more, maybe even a different hormone, you see these conservations of kind of converging on brain regions, converging on molecular neuromodulatory pathways. So there's at least that prima facie sort of analogy that says, okay, there's some utility in terms of understanding the biology of how we are as a social species in an animal model where we can really understand their social biology. Then there's also the sort of the genetic and the sort of circuit aspect of it. And, as we're sort of exploring more and more, we see that there are similar patterns of genetic sensitivity in prairie voles, using the technology that we have, to some of the sort of well-established genetic contributions to different neuropsychiatric disorders in humans. And so again, from biological standpoint, there is the ability to say, okay, at least we're looking at something that may be a really, really significant distillation of how complex social behavior is in humans, but there's some patterns that are emerging in terms of what's happening. And that's where I think the importance of any work in a basic science model is for psychiatry is, it allows us to generate hypothesis, not only about what's happening, but how do we treat, a given set of conditions, right? Whether it's, talking about, the sort of social aspects of depression, social aspects of anxiety. My interest, as I mentioned, is in early psychosis. And, one of the things that we are very, very lacking in terms of treatment in that population is what we call negative symptoms, the social cognition and social components that are impaired in different populations. And so really trying to think about how can we even think about the biology, let alone a treatment for some of these neuropsychiatric conditions. That's where I think a lot of the utility of understanding the basic science is. Mm-hmm. And, historically, psychiatry has, like many other fields of medicine initially, it's been a lot of trials in different patients to see what worked. But more recently, we now have some of the first drugs that are based off of hypothesis-driven basic science. Actually, the first one was actually a hormone-based drug for postpartum depression that was based off of what was known about the biology of parturition, what happened, for women and mothers immediately postpartum, and how does that potentially correlate with what's seen in patients with postpartum depression, which led to the development of a drug specifically for that. So I think that's how sort of the general aspects of thinking about this from a neuropsychiatric standpoint come into play.
And that drug for postpartum depression, what is it exactly, and how does it work?
It's brexanolone, which is it's a steroid hormone analog that is one of the metabolites that changes very dramatically postpartum. And different populations of women postpartum had different drops, basically. And it was noticed that the most significant drop in brexanolone, or it's the hormone analog, correlated for women who had postpartum depression. And so then this analog was developed specifically in this context, and actually, has gone through FDA approval, and now is actually sort of one of the major treatments for that.
Well, how well does it work?
I mean, very well. I mean, no psychiatric medication works as well as some of the other ones, but it actually, it's well above placebo, and it actually, in specific populations of women with postpartum depression, is incredibly effective, right? And it's one aspect of the myriad of biology in this very, very specific, context for women. So, but I think that's actually sort of alluding to why I think the basic science is so important. I mean, as humans, we are very diverse biologically and genetically. And so, the problem in psychiatry is that it's very hard to identify a patient population that shares the same biology, even though they may share the same symptoms. Right? The symptoms are just external, not arbitrary. And I don't in any way want to sort of minimize the importance of really understanding how symptoms present, but the pathways that lead to how symptoms present are very diverse. And so being able to then say, okay, if I take a population that has sort of behaviorally the same sets of symptoms, can I start to understand what the underlying biology is and how it may be different, between different populations, to say a treatment that works in this group of patients may not actually work. And I don't want to have to just try it. I want to be able to say, can I identify who may respond better versus who might not?
Yeah, so in the time remaining, I want to talk a little bit more about your clinical practice. So I think you mentioned that you study childhood psychosis. Can you describe
So I'm not a clinical researcher, but
treat?
the clinic that I'm in is basically we work with kids sort of across the spectrum of various conditions that are starting to show or starting to report what fall into this category of psychotic symptoms.
What is psychosis? That term can trip people up sometimes. So let's talk
Yeah.
about what that means.
So when we use the term psychosis, and I apologize, I'm going to start to sound a little bit like I sound in the clinic. It's a very broad term that has no one specific cause. And so, when we use the term psychosis, it can include things like hallucinations, auditory, visual, actually olfactory hallucinations are the most common. It can include things like paranoid thoughts. It can include things like delusions. It can include disruptions to cognition, so what we call sort of just disorganized thinking. Also, slowing of cognition, blocking of cognition. So these are now some of the things that are in this negative symptom component. It also includes, in the context of some of these illnesses, disruptions to social cognition, either being able to perceive social signals, express them, or sort of integrate them into a social context, right? And the important fact is that you don't have to have all of these. You can have various combinations of them that sort of generally fall into this category of what are sort of psychotic or psychotic-like symptoms. And then the other part of it is, importantly, there's no single cause, right? Many kids actually report these symptoms, and it's part of just natural stages of development. Obviously, as many of us are familiar with, people can have hallucinations of things because of drugs and because of pharmacologic things. Because of fevers, you can have many of these symptoms. And also because of various psychiatric conditions like depression, like schizophrenia, like bipolar disorder. So all of these can actually sort of manifest in different contexts with this general set of symptoms that we call psychosis.
Mm-hmm. And so I guess the real problem is when some of these things are actually showing up at an inappropriate time or in an inappropriate way, in a way that's not typical, and or they persist longer than they might just sort of sporadically pop up for the average kid. Is that more or less accurate?
And the one thing I would also add is when they're disruptive, right? Because
Yep.
it's possible that some of these symptoms can actually occur and they're not disruptive because people have either learned to cope or they're just not, intrusive in a way that actually disrupts their ability to function, right?
Mm-hmm.
And so that's the latter category, obviously, from the perspective of a clinician, that really brings somebody to our clinic, right, to say that this is really interfering with, my child's life or my life, and now we have to figure out how to help them.
Mm-hmm. Are there any, obviously there's a lot of diversity to this, as you said, both in how the stuff shows up and what's driving it. But when you talk about childhood psychosis, broadly speaking, are there any forms of this that you see more commonly, or are there any things that are becoming more common in recent years? And if so, what are they?
So I would say that, sort of in terms of the patients that we see, there is not a single common thing that really sort of unifies even a majority of patients. I will say that there's certain neurodevelopmental syndromes that have a higher likelihood of presenting with psychotic symptoms. So I think, probably the example that's most commonly understood is autism spectrum disorder, right? And autism by itself is an incredibly large spectrum. But what we do know is that where you have children with a diagnosis of autism, there is both genetically and sort of epidemiologically a higher risk for the development of psychotic symptoms. So that's one context in which we see sort of a sensitization. And for the children that do develop them, they tend to develop them a little bit earlier than kids without a diagnosis of autism. But they also share very similar patterns, sort of independent of that. So I think that might be one where you could sort of say, okay, there's a potential sensitivity or sensitization, because of the factors that contribute there. But these symptoms can be reported across the spectrum in terms of early life trauma can lead to this. That by itself is a risk factor for the development of what we call these formal psychotic illnesses like schizophrenia. It can contribute, and there's a whole interaction, like we talked about development and stress and the vulnerabilities of the brain that again. Brings a lot of biology and a lot of sort of human experience to the question of what is causative. And then there's one other thing that I was going to say that I've now kind of forgotten in terms of, and then sorry, in the context of just very complex medical illnesses, right? And when anyone is very sick, you may have had the experience of somebody who's in the hospital who has psychotic experiences, like consistent with delirium because they're just very ill. The same thing can happen when a child is just very sick, either because of a condition or just happens to be very ill for various different reasons, they can develop, because their brains are developing, potential sensitivity to some of these symptoms.
Mm-hmm. Do psychotic-like symptoms, do they happen more frequently at certain like developmental transitions or phases? So like, for example, when you talk about like the onset of puberty, for, colloquially we say, oh, teenagers, their hormones are raging, and we talk about it that way, but your hormonal patterns really are changing. And, we've all had the experience growing up of, going through some of these transitions, and, different aspects of your psychology and your behavior are changing. Do you see sort of transient increases in things like psychotic-like symptoms at certain periods of childhood or adolescent development?
So I'm going to sort of separate that question in terms of when we're talking about a clinical population versus not. So prior to puberty, during childhood, children will like 70% plus of children will report some sort of psychotic, like I hear something or I saw something, right?
Mm-hmm.
It's whether it's part of fantasy play, whether it's, just part of the biology of their developing brain, it's within the realm of normal child development. And, there's no, for the kids that do versus the kids that don't, there's no late life or, later life sequelae. But to the point that you're talking about, what we do know is, for example, what we call these formal psychotic illnesses like schizophrenia,
Mm-hmm.
there is a very significant role for adolescent development. And we assume gonadal hormones because there's a difference between how males and females develop it. The other correlate in terms of the gonadal hormones in particular is there's a second peak for women around menopause in terms of the development of some of these psychotic symptoms or psychotic illnesses. And so the trajectory that we find is that, kids often, and this is actually one of my interests in terms of understanding the social biology, is to identify, are there predictive factors prior to the onset of some of these more common sort of presentations that we associate with, for example, with schizophrenia, like hallucinations or delusions and paranoia, that happen once adolescence starts. And it tends to be that for males, they're a little bit more sensitive earlier, versus for females, they tend to be a little bit offset a little bit later. And so, when you're teaching this, to sort of medical students, like sort of the way that you sort of think about it is, for males, you often see that the sort of peak in these symptoms, even though it's preceded by a couple of years, is right around high school graduation, so around 18. So 16 to 18 is where you sort of start to see the big peak happening for males. And for females, maybe around college graduation, so offset by 2 to 4 years. But then as a consequence of that, and there's a lot of active work trying to understand, is it because it's happening earlier for males that they tend to have more severe symptoms? Or is it that there's actually differences in terms of how the gonadal hormones are working in one context versus the other that causes different severity? I mean, you can imagine the more time a brain has had to develop in the absence of some of these or stresses, the better off it is and more resilient it is. That one explanatory model. We also know that there seems to be an effect of, it's a female protective effect of potentially estrogen and other hormones that may then explain why menopause is the second sort of peak for women in terms of the onset or presentation of some of these symptoms.
Mm-hmm. One last question before we wrap up, because it just occurred to me, and it kind of connects a couple of things that we've talked about this whole time. So you were just talking about menopause, this sort of phase later in life that females have, and we talked earlier about prairie voles and monogamous species and species that have intergenerational cohabitation and alloparental behavior. So, Mike, with all those things in mind, my question is, what is the point of menopause? Why would a species even evolve something like that?
So I think that's an excellent question, and there are many evolutionary theories about the point of menopause, which I certainly don't have my own. I think that one of the ways in which it's viewed is that there is a metabolic benefit potentially to not having the normal reproductive cycle after a certain point of time, in particular where you have the potential for increased genetic sensitivity or susceptibility. But you also then have species that outlive their reproductive age that have a value of sticking around, right? Whether it's this intergenerational providing of care, certainly in the context of more complex species where you have intergenerational transmission of information, which is what we call culture, right? And so the benefits to that and the evolutionary forces that might lead one to the other, I think are very, very complicated. But for example, I think that is one mechanism that may explain some some contribution to why menopause has evolved. And, obviously it's going to be different in a context of all of the associated physiologic changes that are detrimental, right? So then what is the sort of evolutionary cost of that versus, actually just keeping the hormone cycle going, right?
And my understanding is that there's few species where you have female menopause. Humans, I think whales, elephants, it tends to be these sort of highly social, long-lived, multi-generational.
Right. Exactly. And so I think you're highlighting a very important point. Part of it is longevity, but obviously you need longevity to have intergenerational transmission of information. And so, if we can start to tease out, if we have long-lived species that don't enter it, what are the forces that actually might contribute? Another interesting point is, at least when we look at whales and elephants, they're very matrilineal societies, right? Or groups. And so that may be one of the pressures that sort of evolves to sort of, influence some, sort of the development of these physiologic processes.
Well, Dr. Manoli, I want to thank you for your time. This has been fascinating. I could keep going for quite a while on some of this stuff. Is there anything that you want to reiterate that we went over, or anything you want to tie together? We covered quite a bit of ground.
We did. So it's a good question. I think just apropos of kind of like the most recent stuff that we've been doing, I think it is actually a really, really cool time to be doing what we're doing, in part from the genetics, exactly where I come from, but also, thinking about how many different factors contribute to something as complicated as attachment, right? And now the fact that we are uncovering that there is this entire, I don't not to say Pandora's box, but we've lifted up the hood, and I think that there's a lot of really cool stuff that's going to come out of that. In addition to thinking about how experience, one of the things we didn't talk about was attachment is an experience dependent critical period, right? As opposed to many other aspects of development. And so I think that that's going to be a really, really interesting way of thinking about sort of the biology between all of this.
And I don't normally ask guests for book recommendations, but when it comes to this topic, it's so sort of rich and broad. When we think about social attachment and social behavior, whether it's more on the biological side or the psychiatric side or the psychological side, are there any books or resources that you would recommend where people can start to dig into this stuff more?
I mean, to be honest, I actually think starting sort of the original founders of sort of human attachment theory, Bowlby and Ainsworth, they've been, their work is actually incredibly accessible. And then anything that sort of takes that into different contexts. There are many people who have sort of, adapted it, but to be honest, I would actually say some of the original work is worth reading, because they were presenting theories to people that they really had to convince. And so they did a very good job of both presenting the evidence, but also framing it in the context of some of the evolutionary theories that they were basing it off of. And I it's rare that I would say go back to the original, because there are many people who sort of distill it, but I actually think that they're some of the few that really did a good job of presenting those theories.
And what were those names again?
John Bowlby and Mary Ainsworth.
Excellent. Yeah, no, I think that's an interesting recommendation. So again, thank you for your time. This has been fascinating. Professor Manoli, thank you.
Thank you very much, Nick. I really, really enjoyed it.